My name is Julia Salzman. I'm an assistant professor in the Department of Biochemistry, and it's my absolute pleasure today to introduce Professor Bonnie Berger. Professor Berger received her AB in Computer Science, magna cum laude from Brandeis University, and her SM and PhD in Computer Science at MIT while studying randomized algorithms, where her thesis won the George M. Sprouse Prize for Best Research Contribution to Computer Science. Dr. Berger is a professor of applied mathematics at MIT. She is also a professor in the Computational and Biology group at MIT and in the Computer Science and Artificial Intelligence Laboratory, CSAIL. Professor Berger is an affiliated member of the Harvard-MIT Division of Health Sciences and Technology, MIT's Computer Science and Systems Biology Initiative, and has a joint appointment in the Department of Electrical Engineering and Computer Science, all at MIT. She has made many seminal contributions to very diverse fields, many of them centering around applying mathematical techniques to problems in molecular biology, especially in comparative genomics, protein structural motif recognition and discovery, molecular self-assembly and misassembly, as well as functional genomics. Professor Berger has won numerous awards. I don't have time to tell you about all of them, but they include a National Science Foundation Career Award, a Radcliffe Bunting Institute Science Scholarship, Biophysical Society's Dayhoff Award. She was selected for Technology Review's inaugural TR100, has been elected a fellow of the ACM, and received the Recon Test of Time Award in 2011, as well as the NIH Margaret Pittman Director's Award. She is a member of the American Academy of Arts and Sciences and recently received an honorary doctorate from EPFL and was elected a fellow just this year in 2016 of the American Institute for Medical and Biological Engineering. She's currently the vice president of ICSB and on the NIH NIGMS Advisory Council. So without further ado, I'd like to welcome Dr. Berger.
So I noticed in the description that it said I was going to be talking about applying mathematical techniques to problems in biology. Well, I'm going to be speaking about compressive genomics, scaling faster than light. The mission of our field is to address biological problems by using computation in support of or in place of laboratory procedures, with the goal being to get more accurate answers at a greatly reduced cost. We are currently generating massive data sets, as you know in this seminar, so massive that without smart algorithms, we can't effectively analyze them to discover patterns that might provide clues to the underlying biological processes. And there's a big challenge here. The sizes of these databases are growing astronomically. The good news is we have lots of data in which to look for patterns and signals. The bad news is the problems threaten to become computationally intractable due to the sheer enormity of the data. As I will talk about today, by taking advantage of the unique structure of biological data, we will be able to develop algorithms at scale in large-scale genomics, personal genomics, and chemogenomics. Now, given that I'm here and Peter Kim is sitting right in front of me, and we're talking about data, I thought I would do a little walk down memory lane. So back in 1995, when I first started working in this area, GenBank only had 500,000 sequences. And there were only 3,800 protein structures in the PDB. Remember that? And SwissProt only had 50,000 sequences. So this was about the time I decided I would enter into the field as a mathematician and computer scientist. And the dean, Bob Berger, put me together with Peter because I said I wanted to work on protein folding. And Peter posed the question, can we identify coils from just the sequence data? And after many meetings with him teaching me biology, including what an amino acid was, and me teaching him algorithms, I hope, we were able to solve this problem. And we were able to use pairwise residue correlations to identify coils from sequence data. And we wrote these programs, pair coil, multi-coil, learn coil, which to my great surprise as a mathematician, were actually used by thousands of people. I'd never seen anything like this. So that was my first entrance into the field of computational biology. And as you know, our data sets have been growing astronomically. You must have seen this graph before. So here we have genomic data in orange on a log scale. And it is true that computers are getting faster and more economical. And in fact, the amount of computing power is growing by roughly a factor of 2 every year. So the amount of processing we can do per dollar of compute hardware is roughly doubling every year, and that's Moore's law. And back in 2002, this was roughly enough to keep pace with the growth in genomic data. But look what happened after 2004 when the first human genome was sequenced. Genomic data has been going up by roughly a factor of 10 every year. So the way that this has been dealt with in the past is to assume that future computers would be fast enough. But as you can see here, this is clearly not the case. Also lagging is storage, shown in green here. It too is growing exponentially slower than genomic data. So we can't process our data, and we can't store our data, and we're already having major problems transmitting our data. So the only thing that can address this, in my opinion, is to develop fundamentally better algorithms. In fact, we need algorithms that break what we think are the computational limits. We need algorithms, this is from Star Wars if you didn't notice, we need algorithms that are so fast that they scale sublinearly in the size of the data. And that's what we do. We design algorithms to do these kind of computations a lot faster and more space efficiently so that their cost doesn't explode as the size of the databases increase. And in this talk, I'm going to be speaking about sublinear time algorithms, but we also achieve sublinear space. So another thing we do is we take advantage of the unique structure of biological data to be able to come up with novel biological insights from this data. And if you're wondering what this structure is, I'm about to tell you. So others have taken advantage of the structure of biological data in a variety of contexts. There's dimensionality reduction, which you've probably heard of, PCA, principal component analysis, but that is lossy. We're going to mainly focus on lossless today, but we'll deal with a little bit of lossy data analysis toward the end. There's locality-sensitive hashing, which doesn't take advantage of the structure of data per se and can require lots of random hash functions. There's indexing and K-mer-based approaches, which are really good for finding exact matches. But when you try to find inexact matches, your seeding is quick, but then you have to extend, which takes a lot of time for every possible operation. There's compressed sensing, low-dimensional polytopes. But what I'm going to talk about today is an entirely different paradigm for the structure of biological data. That is, we're going to combine both redundancy in the data and its low fractal dimension to be able to develop algorithms that scale. And I'll explain to you in a moment what I mean by that. So the first thing we realize is that there's lots of redundancy in biological data. Okay, so here you're looking at a subtree of the fly phylogeny. And the size of each individual data set is colored individually relative to the other sizes. Okay, so if you look up here, D. Shishleya, the size of it is in gold, and it's nearly identical to D. simulans. So the sizes of the bars are similar. And we color the amount of non-redundant data in black. So again, looking up here, since D. simulans and D. Shishleya are nearly identical, then the amount of non-redundant data or unique data between them is roughly a little more than half the size of D. Shishleya. And here you can see the amount of non-redundant data between these 3 fly genomes is about half. So we decided to try to take advantage of this redundancy. But first, let's view redundancy through a different lens. Let's say we take all the genome sequences in our database, and we try to visualize them in some vector space. Now, this is going to be a very high-dimensional vector space. So the first key observation we made is that our data, where each genome is a point, takes up a very small amount of the entire inhabitable space. And note that this is not a 2D projection of the data. This is just a cartoon. It's just a 2-dimensional cartoon. So another thing that we noticed is that we can cover our data with a relatively small number of small covering spheres of a given radius. And they're shown in like faint gray here. And the reason that this is good is because it allows us to potentially search our data more efficiently. But let's think about why we can do this. If you think about genomic data, it's under pressure from evolution. So you can think about this as an evolutionary tree. And throughout the course of evolution, we explore some branches, but not others. But we don't nearly come close to exploring the entire inhabitable space. And as you're going to see as I go through this talk, this is true for other sorts of biological data too, which of course are also driven by evolution. So it turns out the number of spheres that it takes to cover our data is mathematically defined as the Kolmogorov metric entropy, or metric entropy of the data. And that was brought to our attention recently by Terry Tao. So that's the number of covering spheres. Now, suppose that we want to do a search of our data, and we have a query, which is the green triangle here, and we want to look for it, and we're given some kind of similarity threshold, let's say shown in red here. So we want to find all the points that are within the radius in the red circle related to the query. Now, the problem is that if our data is very congested, then we may have to search a lot of neighboring spheres to find the points of interest that are within the search radius. But as it turns out, biological data has this very nice property that it has what's called low fractal dimension. And what that means is that when you look in the neighboring spheres, there are not too many of them that you have to look at. And in fact, for biological data, fractal dimension is typically 1 or 2, which means that the number of points you have to, or the number of clusters you have to look at scales by 2 to the 1 or 2 to the 2 rather than 2 to the d, which is the dimension of the data and can be really high. So this defines our nice entropy scaling framework. And then we asked ourselves, what can we do with this? Well, as I alluded to a moment ago, we can do search. So let's view search as a bipartite graph, where we have the queries, and we have a database. And the typical search problem, let's say for genomic sequences again, is to match a query to those sequences in the database that it's close to, that are similar to it. And we're usually given a similarity threshold in which we want to find all the matches within that similarity. Now, standard algorithms would look through all the points in the database to find those that match the query. But we realized that because of the redundancy in the database, we can use our entropy scaling structure to do the following. We pre-process the data in the database, which is pretty much fixed. So that's a single pre-processing step. And we group it into clusters based on the similarity of the points or the sequences in the database. And note that these Voronoi regions are our clusters in space, and they're exactly the same as the spheres from the previous slide. So we know that there are entropy scaling, metric entropy number of those spheres, because that's how many spheres it takes to cover our data. And that is what's going to be our compressed database. We're going to pick one representative from each cluster, and what we're going to do then is that'll represent our compressed database, and then we'll do a coarse search at a more relaxed threshold over only the compressed database, over only the cluster representatives, and then we'll do a more fine-grained search on those clusters that we hit. So we're given the fine search threshold by the user, but you would ask now, how do we pick this coarse search threshold to get this to work? Well, for that, we're going to use the triangle inequality. Let's blow up the region of interest, and then note that the query is at most the search region of interest distance away from some point that matches it. And that's the fine search threshold. And then a matching point is at most the cluster radius away from some cluster representative in our compressed database. And I was supposed to be drawing lines. So by the triangle inequality, we have that the query is at most bounded by the fine search radius plus the cluster diameter, the cluster radius. And that works for most things. In fact, it works for distance metrics. Now, the little problem with biologists is they like to use these things called e-values for similarity, and we like to stick to their language, and we like to use what they use. But e-values are not a distance metric. So in that case, they're close enough. So, and I can prove to you, although I'm not going to do it, that if most of the points or the distances between the points obey the triangle inequality in our data set, then in high expectation, we can find all the matches, or nearly all the matches. And for the case of e-value, usually just doubling the radius of the coarse search space will solve the problem. So in practice, we can easily get around that. Now, a lot of the other problems I'm going to tell you about today do the points and the distances between them, which is usually taken from the problem application, they do obey the triangle inequality. So what's the runtime complexity of our algorithm? Well, we pre-process the database, and we can amortize new sequences that come along, and so we don't pay any more for that. We can kind of fit them into our nice entropy scaling structure by putting them in a region where they belong or expanding a cluster as they come along. So what we're going to do is look at the runtime for the search. And as we said, the coarse search is over the compressed database, which is the number of covering spheres, and that's the metric entropy of the database. And the fine search scales with the fractal dimension of our data set. And of course, we have to output our results. So given that fractal dimension is typically 1 or 2, then the metric entropy dominates this runtime. And we can say that the runtime is linear in the metric entropy of the database. Now, we can use a similar argument for space, which I'm not going to do here, but we use instead information theoretic entropy rather than metric entropy. And we get space linear in the information theoretic entropy of the database. Okay, so that's enough for the math. Now we're going to talk about the applications. We're going to try to use this entropy scaling framework to solve a whole bunch of problems. We're going to first focus on large-scale genomics. And this is kind of chronological here. So given the growth in sequencing data that I talked about, we have to wonder if even our fastest algorithms can keep pace. And so Michael Boehm and I, who's actually going to interview here in your department in a week or two or something like that, and I were sitting around and wondering, how are we going to deal with all this data? And we realized that due to the redundancy, that maybe we could compress the data based on the redundancy and try to operate directly on the compressed data. And we call this compressive genomics. So notice that in the original scenario at the top, people would compress the data and then decompress it to operate on it, and that took a lot of time. But with compressive genomics, we just compress the data once and operate directly on the compressed data without significant loss of accuracy in our results. So here's a program that we came up with originally, and it appeared in Nature Biotech 2012 with the compressive genomics paper. And what we did was we compressively accelerated BLAST and called it KBLAST. So here we have runtime in seconds, and here we have 36 yeast genomes. This is just an example. We ran it on all sorts of other genomes and got similar performance. And this is the runtime of straight old BLAST with error bars because we ran on multiple sets. And it's linear in the number of genomes. Whereas this is the runtime of the coarse search only of our algorithm in red, and here's the runtime of the full compressive algorithm in blue. And we were able to find over 99% of the hits that BLAST found. And I know this doesn't look quite constant and sublinear. This one does, but this one doesn't. But as we simulate and add more genomes, it actually does flatten out, as the theory says it should. So we also did this for BLAT and got very similar performance and very similar accuracy and runtime, and as you can see here and here. And we called that KBLAT. So we also did it for protein sequences too, but I'm not going to go into that. And we noticed that our algorithm scaled sublinearly with very high sensitivity on real genomic data. But in some sense, this was easier than some other problems that we were going to encounter. And this fit very nicely into our entropy scaling framework, but historically it came first and got us to come up with the theory that I already told you about. And here we were basically thought we were just using redundancy, but in some sense the reason it worked is that we were using low fractal dimension as well. So we've shown how to use the structure of the data to do large-scale genomics. Now let's take a look at personal genomics. And these are just my names for these things. And in particular, we're going to look at compressive mapping, which even when the first paper came out, it wasn't clear how to accelerate mapping, because mapping's a little more tricky. And I'll explain why. So the goal of personal genomics is to sequence your genome. And I'm sure you know how this works, but I'll remind you, the way this is done is you take many copies of an individual's genome, and you cut them up many times at random, which is known as shotgun sequencing, and they create fragments. And some of the locations are covered by multiple fragments. And then if you're, for instance, using Illumina sequencing, you can read 100 base pairs or so at a time from one or both ends. And so to read a genome, we're given short, error-prone reads, although with Illumina not so error-prone, from random locations in a genome, and we want to use those to piece together an individual's full genome. So if we're interested in some disease, let's say breast cancer, we might also want to know where your genome differs from someone else's. And those locations are called single nucleotide polymorphisms. So the way this is typically done is through what's called a next-gen sequencing analysis pipeline, or a sequence analysis pipeline. We're given raw reads from a single or many individuals, And then we use an existing mapper, let's say Bowtie2 or BWA, and we use these to map onto some reference genome. And then once that's done, we do what's called variant calling. And these are the popular tools, GATK or SAM tools, to tell the places, the actual locations, and what base is there where your genome differs from someone else's. So there are all these projects out there, like the 1,000 Genomes projects, where people have donated their genomes and made them public, where people can, we can collect all sorts of genomes that we can do analysis on. And so what we want to do is we want to leverage the information across all these genomes, that is the variation, to come up with novel associations with disease. So that's what mapping is good for. And as it turns out, and the whole sequence analysis pipeline, as it turns out, this step with Bowtie2 or BWA is the most computationally intensive step in this pipeline by far. And that's when I'm going to show you how to compressively accelerate by orders of magnitude. So remember, we have our bipartite graph. This is basically a search problem. We want to map reads to, those are our queries, are the reads. And in this case, we're mapping them to loci in the reference genome. And we want to map a given query, let's say, to those places that it matches in the reference genome. We've already seen this problem. However, that's the punchline. But in this case, before we compressed the database for search. But the hard part about doing mapping was we didn't know what to do with all the reads because we can't pre-process those. We don't know those ahead of time. They have to count in our runtime. So we also realized that if you're mapping reads from 1,000 individuals to a reference genome, you're going to be doing trillions of mapping calls. And that's just not at all computationally feasible. So we realized that the reads actually had more redundancy than even the reference genome or genomes. And so we realized we could also cluster the reads based on this redundancy. Now, before I tell you about that, I want to let you know that people kind of are interested in two different kinds of mapping. There's something called best mapping, which maps a query to the best match in the reference genome. And that's what everyone has been using. And the reason is because that's the only thing that was tractable. And then there's something called all mapping, where we map a query to all the matches within a given similarity threshold in the reference genome. And that's what we really want to do, because if we can do that, all mapping, or what we call multi-mapping, or map it to lots of locations, it doesn't have to be all, that gives us much better downstream analysis, like much better variant calling. And Steven Salzberg wrote a review article on this, and that's what he said in the review article. Now, I'm pleased to tell you that they are starting to use all mapping based on our work in the PanCan pipeline and also in GATK. But before that, people didn't think it was tractable. So how does this work? Well, we do our same game of clustering the reference genome, and we have our entropy scaling structure, and then we can search through only the cluster representatives in a coarse search. Now, in this case, we're looking at k-mers. Our points in this case are k-mers that are 32 to 63 base pairs long, and there's a good reason for this in terms of uniqueness and redundancy, and we've actually analyzed what the right k is. But so we're using k-mers in the reference, and we're using k-mers in the reads. And they're 32 to 63 base pairs long. And not only do we cluster the reference k-mers, but we also cluster k-mers in the reads by their similarity, as I said. So these purple ones all cluster together. So then we only need to take a k-mer representative for the read and it came a representative for the reference, and map between them. And that's our course mapping stage instead of our course search stage. And once we do that, and let me explain that all our software is written as wrappers to existing software. So we basically just come up with this course representation, and we can run Bowtie 2, we can run BWA. We take standard formats, and we just give those pipelines. It fits into any next-gen sequencing analysis pipeline, no problem. So we're basically using, for the course mapping, BWA or Bowtie 2, but on a much smaller data set, which is where we get our increase in time. And then, our decrease in time. And then to do the find search, to look in the clusters, we only store the differences from the points in the cluster to the cluster representative. We need only store the differences. So the clusters are compressed as well. And to do the find search, we just merely trace simultaneously between those reads that have been grouped together in the same cluster and those places in the reference genome that are similar. Or we trace through the differences. And this paper will appear on April 7th in Nature Biotechnology, coming up. And it's with Dennis Yurikoglu, William Yu, and Zhenpeng. And the program is called Cora. So Cora is our multi-mapping or all-mapping read mapper. And so how does Cora do? Well, we tested it on the 1,000 Genomes project. And the red bar is the runtime of Bowtie 2, straight up. The purple bars are the runtime of BWA. This is runtime in hours. So you notice that Bowtie 2, we had to basically conjecture how it ran because it just took way too long. And we tested it on the FIN1, FIN2, and FIN4 data sets in the 1,000 Genomes project. So FIN1 is a data set that has 4x coverage, which means that every location in the reference genome is covered by roughly 4 reads. And FIN2 has 8x coverage, and FIN3 has 16x coverage. And as you can see, as the amount of coverage increases, our gains, which are shown, I don't even know if you can see them, our runtimes are shown in orange and aqua here at the bottom. And our gains become much greater as the size and the coverage of the data sets grows. And this is all mapping with indels, with insertions and deletions. And that's what people really want to do. We can go up to edit distance 10, in fact, and not lose much in runtime. And we can go up to edit distance 4 and lose nothing in runtime. But there's another program out there that I should mention that does all mapping with Hamming distance, and that's called Mrs. Fast Ultra, shown in taupe here. And we're 6 times faster than that if we don't sort their output. They output not in the same order as the reads that are input. If we have to do the sorting for them, we're 10 times faster because our output comes out in the correct order, but theirs doesn't. And then we can say that our algorithm, Cora, scales sublinearly with very high sensitivity on real genomic data because, in this case, we basically are in the high 99%, similar to Mrs. Fast Ultra, and we get very near the same accuracy as Bowtie 2 and BWA. And for FIN1, we're 46x faster. And for FIN4, we're 56x faster. And that just goes up. OK, so now let's turn to another problem in personal genomics, what I call personal genomics, just to structure the talk. And that is metagenomic analysis. So we also can speed up metagenomic analysis. So we've heard a lot lately about how we're not just our DNA, but we're what's in our gut. So metagenomics is a study of bacteria or whatever in an environment. And in this case, we want to know what is in the gut. We want to sequence what's in the gut. And we get something like this, which shows kind of an evolutionary tree of the sequences that are in your gut and how they relate to each other. And the reason we want to do this is there's lots of reason to believe it's linked to various autoimmune diseases, for one, and other types of diseases. So we want to kind of sequence different people's guts, and there's a lot more data because we're talking about bacterial genomes, yeast, fungi. We have lots and lots of genomes in this case, and they're all very different than each other. So it's in some sense a more high complexity problem. We're not just dealing with one reference genome. So one thing we could do is use our core-up mapper that we just spoke about. We could map the DNA sequences that we get out of your gut to a whole bunch of references. And that will tell us, which ones we hit and what's present and where we hit them. However, what people typically do right now is they convert the DNA into proteins, and they use BLASTX, which maps DNA to proteins that are, in some sense, a proxy for what's in the gut, because it takes a lot less time and it's quick. So they use BLASTX, and given reads from a microbiome, what they do is they match each read with similar proteins. Another thing they do is abundance counting. They want to know kind of how much of each kind of type of genome you have in the gut. And we work on that, but I'm not going to talk about that today. But we have an algorithm coming out in Recom 2016 on that. So we came up with this program, MICA, and how does it do? Well, here's running time, but this is going up by a factor of 10, unlike our other running times. And first, we checked out how BLASTX did, which is what all the microbiome sequence analysis pipelines use. And that took 58,000 minutes to run on one single human gut microbiome. And Diamond, which newly came out, and it was state of the art, takes 54 minutes. And our MICA takes about 15 minutes. And in fact, if you look here, it's a 3.5x speed up. Oh, I guess it's a 3700x speed up over BLASTX, MICA. With 90% sensitivity, we can get like 1600x speed up at 95% sensitivity. So there's a trade-off there. And we get 3.5x speed up over Diamond. And now we can get that to 4x with the new microbiome databases. So we're even able to speed up the state of the art there by using our entropy scaling framework in a similar way. So we've seen how that works for actual sequence genomic data. Now I'm going to turn to a different type of data. I'm going to turn to small molecule search. So it's a very different kind of thing, but it turns out the same framework also works. So when we first started in 1995, PubChem didn't even exist. But it came along in 2004, and it too, if you look at millions of compounds in PubChem, they have also been growing very, very quickly. So we would like to be able to search PubChem. Now, it turns out that the program used to do that by everybody is Janet Thornton's SMSD, but you can't even search for a single molecule on a lot of different types of computers, only on very, very advanced computers, quick enough to be able to do that. So if you try to search like on your just basic laptop computer with SMSD, you can't do the search for even a single molecule. It takes too long. So what do we mean by these small molecules? Well, here's an example of one. It's called zanamivir, and it's an antiviral that binds to the neuraminidase protein of the influenza virus. And we're very interested in these kind of compounds because people want to do what's called rational drug design. That means they want to look for small molecules that bind a receptor, and they want to design small molecules that bind a receptor, which is what Peter's trying to do for HIV right now. But it's really, really expensive to do this. So what they do is they scan these public chemical compound databases to find ones that may behave similarly. Or they might want to find a drug like zanamivir that doesn't have bad side effects, whereas one of the drugs does. Okay, so this is why we would want to search these chemical compound databases. And I'm going to show you now how to do it very quickly. So again, we're going to use the structure of the database. But in this case, our points are going to be the small molecules. And I'm going to show you how to abstract them to get the cluster representatives. And the distance between them is going to be the same distance that Janet Thornton uses in SMSD. Basically, we take our distance metrics from the program that we're compressively accelerating. We don't have to come up with a distance metric. So we use Tanimoto distance. So what we do here, it's really easy. Here's an example. We have caffeine as a small molecule. We have adenine. And all we do is we map the atoms to become the nodes. And the edges in the simple cycles become the edges. So this is an abstract representation or a cluster representative for these two molecules. And interestingly enough, theobromine, which is the active component in cocoa, is also in this class. And surprisingly enough, by using this as our coarse compressed database, we are able to search only over these abstract representations very efficiently. And then we store the differences between this and the small molecules inside the cluster. And we get those back through the find search. So our program's called Amylite, and it too was in the entropy scaling search paper, which just came out in Cell Systems in the fall. And so how do we do? Well, we looked at our speed up versus the Thornton Labs SMSD. Here's the factor of speed up that we get. And we looked at database sizes of 1 million, 10 million, 47 million chemical compounds. And we found that on average, we get an order of magnitude speed up. And that will only increase as our databases grow. Okay, one last thing. I've been talking about lossy search, or lossless search. I've been talking about lossless search, pretty much lossless search. There's also the storage problem. And I said that we can get lossless storage sublinearly amount of storage for the problems that we were considering. But there's another problem. Not only does the read data take up lots of space, but these FASTQ files that people pass around are too big to transmit because they also have these quality scores, which take up even more space than the read values. So we had a paper in Nature Biotechnology in 2015 that showed that if we compress the quality scores in a certain way, and it's very similar to our entropy scaling framework, then we get surprisingly good results. Now, we're part of this consortium, MPEG, the Motion Picture Industries Consortium for Genome Compression now, and they're using our program Quartz, and they're using another program that's developed here in the Weissman lab, and QVZ, I think, and they're trying them out and seeing how well the data is compressed and what works well. So we came up with this Quartz program, and what it did was it basically used read data that we'd seen before as a prior for smoothing the quality scores. So we essentially throw out the quality scores when we have a lot of confidence in them. When read data we see it's covered by a lot of read data to back it up. So I've already talked about this. And so here's our typical next-gen sequencing analysis pipeline. And as usual, we just plug in our quartz algorithm to get compressed quality scores. That is, we smooth the ones that we're sure of based on reads as priors, that we've seen a lot of reads covering a given location, and we feed it right back into the regular pipeline. And all our tools work this way. We just have a wrapper, and we feed in a smoothed FASTQ file. And lo and behold, we get orders of magnitude speed up, and for some bizarre reason, which I actually understand somewhat, we got better variant calling. So our performance using a pipeline that used both Pi2 for read mapping and GATK for variant calling, our quartz AUC is in red, and just looking at uncompressed data is in blue. And we even did better for some reason, I think because we smoothed away all the noise. And in fact, the punchline is we actually discard 95% of the quality scores and get improved performance. So we don't even need them. Although if we discard all of them, we get much worse performance. So we can't do that. We have to be clever about this. So I'll wrap up. We have a whole bunch of tools available. These are our compressive genomic search tools. They're at these various websites. The Kablast ones and Kablat are here, CAST. A lot of the tools are at GEMS. Cora is our read mapper. And we also have our compressive storage quartz tool, which is lossy compression. And we've also applied this, as I said, to non-genomic data, well, non-sequencing data. Small molecule search I talked about. Amylite is at the GEMS website. And we've also, I won't have time to talk about, but we've applied this to protein structure search. We can speed up Rachel Kolodny's FragBag using similar techniques and do very fast protein structure search. So to conclude, I want to thank all the amazing people that worked on this. William Yu and Noah Daniels were first author on the entropy scaling search paper and the applications. Dennis Yurikoglu is first author on the Cora mapper. David Denko was an amazing undergrad who helped us a lot with the different applications like metagenomics and PubChem search. Michael Bayman-Peru and I came up with the original compressive genomics paper in Nature Biotech in 2012. And Zhen Peng has been a big help on many of the cell system and the, not the cell system, he was a big help on the mapping paper. And there's my group at Recom.
I think we have a few minutes for questions.
Thanks for coming.
Okay. I had a, oh.
So I just wanted to know, did you run your comparisons with the existing read mapper single-threaded, or in fact, can you run this fast search in a distributed setting?
Yes, we can run it in parallel, but we did not even, and we still got those improvements.
Okay.
And that counts the clustering of the reads. Hi, Michael.
Bonnie, you showed that picture at the beginning where you sort of showed space decomposes really nicely.
Yes.
Doesn't that lead to enormously big advantages in parallelization as well?
Exactly. That's actually very true, and we're looking into that. But it should be quite easy to parallelize it. Good point. Yes.
Hi. I was wondering, in the Cora workflow, did you look to see how that affects the downstream variant calling? Because I was thinking that maybe there's a slight loss in sensitivity. Maybe that's in places where there's like rare complex structural variation that the seed and extend method
Well,
would
it
be
turns
more
out
apt
we
at
even do
applying.
better because we do all mapping, and we did have to compare to all sorts of pipelines and the downstream accuracy, and we were extremely close, like 99% kind of similar, and that's also in the paper.
Did you characterize though, like the variants that you're missing, are they rarer? Is there any relationship?
Well,
Or more
we
complex?
haven't, we're working on that with the GATK team at the Broad. That's our next step. But I would think that we'll be able to find more interesting ones because we do an all mapping rather than a best mapping. Well, all we need is one hook into the reference genome, and we have this really cool indexing structure for the reference genome, which can be used for all sorts of other purposes. And it doesn't even have to be a best hook. It just has to be any old hook, and then we can kind of recreate all the things that are similar. Thanks for the questions.
I had a question, actually. So you showed incredible performance of your algorithm on the 1,000 Genomes project with Illumina reads, and I was wondering if you've tested its performance on PacBio reads.
Okay, so that's a whole other thing. I'm sure the longer reads will be extremely helpful in terms of runtime, but they have a lot of errors in the PacBio reads. And until they get their errors better, or Illumina goes to 700 base pair reads, although they're claiming they're going to keep their technology the same for the next 2 or 3 years because it's selling so well. But eventually they do want to get up to 700 base pair reads. Until PacBio improves its error rate, I wouldn't use it for variant calling per se without some backup. Now, one thing that could help us is we're thinking of applying our quartz algorithm to the PacBio read data to be able to detect errors in that data. But just a thought. Thanks for coming.